Wonderful. So it's so nice to see so many people on Monday morning, and on a freezing Monday morning. So it's my name is Johan Sabate. I'm a professor of physics at the Department of Physics and Astronomy. I also have a courtesy appointment at the chemistry department. My guest today is Professor Eric Betzig, the winner of the 2014 Nobel Prize in Chemistry, together with Stefan Hall and William Morner. Professor Eric Betzig is Professor of Molecular and Cell Biology, the Eugene Cummins Presidential Chair in Experimental Physics, a Senior Fellow at the Janelia Research Campus, and an investigator of the Howard Hughes Medical Institute at the University of California, Berkeley. Eric is, as I said, the 2014 Nobel Laureate in Chemistry. This lecture is special because it's co-sponsored by 3 departments in campus: the Department of Biochemistry and Molecular Biology, Department of Chemistry, Department of Physics and Astronomy. The lecture is also very special. It's a signature lecture. Provost Jack Hu describes the signature lecture as, I quote, "Signature lectures provide an opportunity for the university community to engage with nationally renowned speakers and learn from their wealth of knowledge and insights. This lecture series enriches the UJ learning experience by fostering the open exchange of ideas and deepening of our interdisciplinary knowledge," end quote. And all these signature lectures are free and open to the public. Before I briefly introduce our distinguished guests, I have a few people to thank, without whom this extraordinary event could not have been possible. The Provost Office, and particularly Provost Hu, for selecting Eric's talk as part of the Distinguished Lecture Series. So let's give the Provost Office a warm thank you. The various chairs of the departments for helping with organizing this event: Professor Tina Salguero, Associate of Chemistry; Professor Craig Wiggert, Head of Physics and Astronomy; Professor Chris West, Head of Molecular and Cell Biology. So let's again give them a warm thank you for the help. And I have also a special thank you to Professor Alan Dorsey, our former dean, who really helped out organizing this event. So Alan, thank you so much for all the help. All right, now back to our truly distinguished guest, Professor Eric Betzig. Eric's research and impact is extraordinary in the following 2 senses. He pioneered 2 exclusively and also complementary research fields. What's now called more broadly as a nano-optics or near-field community. He is the one who began that in the modern sense with his thesis. Basically, I'll tell you a little bit about that. And then later on, he used those techniques in a profound way, in a very new way to do biology, to understand biology, which he will tell you a lot about. So the material world, from the cell phone you use to the car that you drive, is driven by understanding materials, using materials to do stuff. And for that, one needs to know the physics or the chemistry of things. And for that, one needs to see how things work. And near-field optics allow you to do that. And Eric was one of the first people who actually built these kinds of instruments. Now, this field has grown quite a bit. It comes with different names, the near-field optics group or nano-optics and so on. But he really was a pioneer of the field. And then later on, as I said, he used those to do biology, to apply these techniques to super resolution imaging and spectroscopy using all kinds of techniques. And in another sense also, his path to these pioneering fields was also extraordinary. The way he got there, how his scientific journey. He probably will talk a little bit about that. But he got his undergraduate degree at Caltech and then went to Cornell to do a PhD, where he actually did a thesis on this new field at the time called near-field optics. And then he went to continue to work on it, went to Bell Labs, and expanded this field, wrote pioneering papers on it. But then he got tired of it, and he was exhausted of academia. I think he has a beautiful, very unorthodox history how he actually moved from one place to another. He left Bell Labs, and then he actually became unemployed, self-unemployed, and a homestay dad, and for several years actually, and left science completely, right? And this is an amazing story. And then came back to science. He worked with his dad in a company which turned out to be extremely, I don't know how many, he sold several microscopes. He sold two microscopes, two of some kind of thing, and then went back to science again. Then what makes the story very interesting and extraordinary is he had an idea that became fruitful under some circumstance, he would probably tell you, and built the first super resolution microscope with his buddy, tennis partner and best friend Harald Hess, in his living room. So a lesson to learn here, how to structure your homes for science. And that work actually led to the Nobel Prize. And the rest is history. He went on from there to Janelia Lab, and then now he's at UC Berkeley. There's a long, very, very exciting history how to actually he succeeded and what he did. So another thing, as I mentioned, the Nobel Prize was given for his work that he built this microscope with Harald Hess, super resolution single molecule localization microscope in his friend's living room, and for that he received the Nobel Prize in chemistry in 2014. And just recently, I have to mention this I just found out just Googling around, Eric and his conspirator in crime, his tennis partner, as I said, Harald Hess, they were both inducted into the 2024 class of the National Inventors Hall of Fame for their invention of the photoactivated localization microscopy. So let's give him a very thank you, Eric. So with that, I think I'm going to leave the floor for Eric, and thank you so much for accepting this invitation.
Yeah. Okay, thank you, Johannes, Lisa, and all the other organizers, particularly Johannes. He knows I hate to give talks, and I very rarely do, but man, he is one persistent man. Anyway, so microscopes have been around for 400 years, and for the first 300 of those years, they were the primary tools by which we were able to understand living systems below the level of the naked eye. Microscopes gradually improved over the next 2 centuries until at the end of the 19th century, a microscope manufacturer, Carl Zeiss, frustrated with the irreproducibility of his making microscopes, recruited a physicist, Ernst Abbe, to try to understand how to make a theoretically optimum microscope, and Abbe discovered that there was a limit to how small you could see that was roughly half of the wavelength of light. And for the next 100 years, that was it. And so therefore, the microscope that you could buy in 1880, using Abbe's principles and Zeiss's manufacturing capability, was conceptually no different than the microscope you could buy in 1980. Sure, we now had cameras and better detectors and many, many other things, but the basic functioning was exactly the same. So as a result of that sort of stagnation, different tools had to come to the fore to push biology forward. Those were biochemistry, molecular biology, and structural biology. And these have been so successful that they've become the dominant paradigms by which most biologists understand the cell today. But I would argue that these techniques have a fundamental problem, and that essentially they're all reductionist. They're all about grinding the cell up into parts and looking at the parts. But the cell is a dynamic, complex system. So trying to understand something as complex as the cell is like trying to take a random pile of engine parts and then reverse engineering the operation of the car engine. It's not going to happen. The only way you can really understand the totality of the picture is by learning what you can about the parts and how they work, but in the end being able to see how these parts work together as a complete system. And light microscopy is the only tool in our toolbox that can do that. Electrons can't do that, X-rays can't do that. The only way we can look at the whole system at high resolution is with photons, visible photons. Now, the good news is that starting in the 1980s, different, diverse technologies came together unexpectedly to ignite a revolution in optical microscopy. First was the invention of the transistor in '47, which led to integrated circuits, and by the time I was in graduate school, PCs that could actually automate the operation of microscopes. The similar technology ended up in CCD sensors, which made very sensitive detectors to be able to then take digital images that you could then do analysis on of what you're seeing in the microscopes. And so by 1980, with digital interference contrast, people were now able to capture images of beautiful dynamics of cells using that technology. In the 1980s, although fluorescence has been around forever, its application to be able to specifically illuminate different subcellular compartments became very refined by the 1980s, so that you were able to see individual subcellular components in live cells. And to be able to excite that fluorescence, around 1980, lasers were now finally cheap enough to go in individual labs so that we could look at the fluorophores and excite them very carefully. And then later on, as we moved into the 21st century, use the coherent properties of that laser light to sculpt the light that we're shooting at the samples in various interesting ways. So let's see how we got to where we are today. Well, we start with the diffraction limit. So what does that mean if we have half a wavelength of light of resolution? How limiting is that? Well, if this represents the size of a protein molecule, then that fuzzy blob is what it would look like at the diffraction limit. So there's about a 2 order of magnitude disconnect between where we are and where we'd like to be to see those fundamental building blocks. So as Johannes mentioned, after undergrad, I went to Cornell for graduate school. And I grew up in the 60s as a child of Star Trek and the Apollo program. And I've always believed in the transformative ability of science and technology to make the world a better place. So I wanted to do something important, transformative. So there was a couple of professors, Mike Isaacson, who was an electron microscopist, Aaron Lewis, who was a Raman spectroscopist, who had a crazy idea to try to make an optical microscope that would have the resolution of an electron microscope and still be able to see living cells. And I said, well, that sounds transformative. Sign me on. So the idea was, with Mike's electron microscope, they could drill holes much smaller than the wavelength of light in an opaque screen. And the idea is if you put that screen very close to a specimen, and you shine light on the side opposite the specimen, the light that comes through the hole has to be initially collimated to that sub-wavelength size. And so now what you have is a little nano flashlight that you could drive point by point across the specimen and get a super resolution image of the sample. So that was called near-field microscopy, and that's what I did for my thesis. And I had some sort of half-assed, tantalizing results, I would say, as part of my thesis, but nothing to brag too much about. But it was just enough to get me into the door to where I really wanted to go, which was, at that time, the preeminent research institution in the world, AT&T Bell Labs. So I got my own lab there. I struggled for several years with the technology, but finally made some improvements. And eventually we had a number of hits. At one time, with using those small little apertures, we were able to write magnetic recording bits about an order of magnitude smaller than the recording densities used at the time, so we had a world record there. We were able to do the first super resolution imaging by fluorescence. This is the actin cytoskeleton in a fibroblast cell. But again, it's a fixed and dead cell because this technique's very slow. And so we had a number of hits. So life was pretty good for a while, but pretty soon I hit a wall. Well, first let me tell you one more thing we did before I hit the wall, which is when we were looking at those cells, we realized that we might have the sensitivity to actually see single molecules. So this was a hot field at the time because everybody wanted to get to the ability to detect single molecules as sort of an ultimate limit of sensitivity. In the year before, W.E. Morner had been able to do this by looking at the spectral signature of single molecules in a system that worked near absolute zero, where these spectral lines are narrow. I was able to do it at room temperature because the problem wasn't so much the signal coming from a single molecule, but the background from everything else around it. The near-field microscope makes it so we're only looking at the light that's coming from the molecule and not the background, so then it became easy. So we were able to detect single molecules. We were able to localize where they were positionally to on the order of 12 nanometers, about a 50th of the wavelength of light. And we were able to even by polarization infer the orientations of their dipoles. In an experiment the year later, I teamed up with my best friend and tennis partner, as Johannes said, Harold Hess, who was doing low temperature scanning tunneling microscopy of semiconductors. We ripped out the STM probe, put in the near field probe, and started to look at quantum wells, which are the semiconductor structures which glow and create the energy in a laser pointer, for example. And they're made of these thin sandwiches of different types of semiconductor materials, which confine electrons in what are called holes in these things called excitons that move in those quantum wells. And we were able to show that the emission that comes out of those wells is not random and anywhere, but instead it's confined to potholes in these little thin films where the roughness changes by 1 atomic layer of thickness. And the excitons get stuck there and then they emit light there. And even with my near field tip, there might be dozens of these excitation sites under the probe, and so not conventionally resolvable. Because we were near absolute zero, the spectral signatures were so narrow that if you built them up as different colors in a three-dimensional space, then you could isolate them and study every exciton site individually. So that's a really important idea we'll come back to later. But my real dream was, again, following what I'd wanted to do in graduate school, which was to look at living cells with the resolution of electron microscope. And by '93, I realized this was never going to happen with near field, because the problem is the light that comes out of that tiny little hole spreads ridiculously rapidly on the other side. And if you're even more than, say, 10, 20 nanometers away from the specimen, you get a noticeable loss of resolution. By the time you're 100 nanometers away, forget it. But cells are not flat on a 100 nanometer scale. And so I was sunk. That was not going to work. So that was disappointing, to say the least, since that was my dream. But I also realized something else, which is that I am not and never will be an academic scientist. One of the things that happened was something I've seen over and over again in science is when you start, everybody says, this is crazy, this is nuts, no photons will go through that hole, this will not create super resolution, I don't believe it, blah, 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 blah. And you give them all good physics arguments for why they're wrong, they don't listen, you build it, you start to gain some acceptance. Eventually there's a turning point, a phase transition. And all of a sudden, everybody believes it. And everybody says, well, of course. And then everybody wants to do it. And they start getting into it. And a lot of the people getting into it don't really know what they're doing. Most of them don't know what they're doing. They're in it because it's hot, okay? And so physics and all sorts of fields of academia are just overwhelmed with fads where things just grow and grow and grow. People, in order to get funding and attention, say all sorts of silly things. And I felt like basically the signal of good work that was coming out of the field was staying constant, and the noise was going to infinity, so the signal to noise of the field was going to zero very rapidly. And I also felt like I'm sorry to say this, but I felt like every paper we did was nothing but the justification for 100 pieces of crap that followed in its wake, and everything I did was a waste of time and a waste of taxpayers' money. And I really felt very strongly about that. And so that, coupled with feeling that I'd hit a technological dead end with the technique, caused me to just quit in frustration, and so I left Bell. And so as Johanna said, the other reason I did this, though, was a final one, is that, I teamed up with Harold in '89. As soon as I got there, it was basically a clear soulmate of mine. And so we became best friends. And, Bell Labs, you go there and there's a 50 years of history of so much great science, and you feel like you don't belong, and you're on probation, and so you have to work. So Harold and I would get into work at 4:30 every morning. If one of us beat us to the number one spot in the parking lot, the other would put his hand on the hood of the car to find out how many minutes they'd beaten us by. And then we would go and we would work until sunrise, and then we would go play tennis for an hour, and then we'd come back and work, and then we'd work till dinner, and then we'd go to the same Chinese restaurant for dinner every night, and then we come back in the lab, and then we work, and then we go home around 10. And then we do this 7 days a week over and over and over again. It was one of the greatest times of my life. Absolutely great. Loved it, but it adds a lot of miles in a hurry. So after 6 years, let's just say I was burned out for more than the reasons of the limitations of near field, and so it was time to recharge and quit. So I quit, and I became a house husband, and we had our first baby in '93. I really love my kids. I have 5 kids. I highly recommend to anybody to have kids. There's nothing more rewarding in life than to have kids and to raise kids. I find it very sad to see more and more people seemingly to go childless when it's really the greatest joy in life. But anyway, so I had the first kid. I was walking the kid around in the stroller, and I wasn't even thinking about near field or anything else. I was trying to think about, how am I going to get a paycheck again? And it hit me that you could combine the single molecule experiment I did with the quantum well experiment that Harold and I did to come up with a different approach to super resolution. So the idea I said is, like I said, by Abbey, those single molecules look like fuzzy balls in a microscope. The problem in a biological sample is the molecules are so close together, the fuzzy balls overlap, and you just have a mess. But what if, as in that experiment where Harold and I did, where we could see the excitons individually by spreading them by wavelength, what if we could spread the fuzzy balls by some distinguishing optical characteristic, that they're different from one another, either they glow in different colors or whatever? Then I can build a 3-dimensional space where now they're separated by color or whatever that dimension is. Now those fuzzy balls are isolated in a 3-dimensional space. I can point to the center of that basket, of each of those little basketballs, with better precision than the diameter of the basketball, and then plot where they are in that multidimensional space, then collapse that back down on the spatial coordinates, and have the location of every molecule correctly, precisely positioned, and now I have a super resolution image without using a near field tip. And so I was really enamored of that idea, but I was unemployed and didn't have any way to do it. So I published that idea just on a lark on my own. See, that's me, Ensom Enterprises. Okay, that's my whenever anybody has an LLC, I usually think that's a synonym for being unemployed. But I've used that synonym several times in my life. But anyway, so I almost didn't publish that paper, but it's a good thing I did, because that was one of the 2 papers the committee cited for giving me the Nobel. Okay. So in the end, I followed my backup plan. So my dad had started a machine tool company in '85, and it grew to about 300 employees and $75 million in sales by the mid-'90s. And he always wanted me to join him in that company, and I knew that was an option. So in the end, not thinking of anything better, I said, yes, I'd like to join you, Dad. So I did it, and he signed me up as VP of R&D, and I wanted to come up with a new architecture for the high volume manufacturing of auto parts, which is what this company did. And at that time, the big 3 was under considerable pressure from Japanese manufacturers to cut costs. I came up with a crazy idea that we use a very old technology, hydraulics, marry it to modern nonlinear control algorithms that I could put into a PC. I made this machine that would move 4 tons at 8 Gs of acceleration and position it anywhere within a meter cube to 5 micron precision in 100 milliseconds. And so it was like a hummingbird go bzz, bzz, cut, 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 bzz, bzz, bzz. It was massively more performative, much smaller because you get power to weight gains out the wazoo, so everything gets smaller, it gets cheaper, it gets faster, it gets better. I love this technology, and I put my heart and soul 3 to 4 years developing it, then went on the road show and tried to sell it, and nobody wanted to touch it. Everybody was just scared to say. I would bring in union guys and I'd hit the go button and they'd jump back because they think the thing is going to run off the rails and crush them. It was just way too different and too much of a risk in a field which was not known for taking risks. And so in the end I sold 2 of them, and it was a major disaster. And so in the end I apologized to my dad and I said, I'm sorry for burning $1 million of your money on this, but this is just not the right life for me. And so I quit again, and again had no plan. So while I was doing that, Bell continued to implode after the Bell system breakup, and Harold eventually left and went to work for a company that made storage devices to measure disk drives for the storage industry. And he was also feeling a little bit itchy about working in industry, and so we would meet at different national parks and just go through our midlife crises together, trying to figure out, what do we want to do, how are we going to make a living, and still not completely sell our souls, and what's important in life, and where do we go from here. So we meet in various parks and so forth, and so that got me to thinking, well, eventually Harold and these talks convinced me that while I didn't like academia and I didn't like the nature of the scientific establishment and publications and how it all worked, I really missed science. I really missed doing science. I really missed being in a lab. And so I started reading the scientific literature, and one of the first papers I ran across was about Chalfie and Gang's green fluorescent protein, where you could genetically modify any protein you want to have a handle on it that would also encode a glowing protein from a jellyfish, so that when you put that in a cell, it would produce copies of any protein you want, now glowing with that label. To me, this paper was just changed my life. It came out the year after I quit, and so my timing couldn't have been worse because I saw it 10 years after it came out. But it was so stunning. In the time I was trying to do fluorescence with near field, the difficulty of labeling anything I wanted to see with high enough density and specificity was even more limiting than the microscope itself. And so the idea that you could use biology to do all of this for free, it was the most elegant thing I had ever seen. I said, God damn it. I knew I didn't want to be a microscopist, but God damn it, because of this, I got to become a microscopist again. So I tried to think about ways of doing microscopy again. I came up with an idea, and it was really, really, really useful to be unemployed in that time because I could go back. I'd forgotten so much of my physics that I had learned over the years while I was at my dad's company. And so I started actually just reopening my old freshman textbooks and problem sets and started doing the problem sets and stuff. And I started to learn electromagnetism far better than I ever did before by learning it on my own at my own pace. And I came up with this idea I knew about standing waves and how 2 beams can come together to create a pattern of light. And I wanted to generalize that to 3 dimensions to see if I could make a multifocal field for ultra-fast, ultra-parallel imaging like a massively parallel confocal microscope. And eventually I came up with what I think is a beautiful theory. It's probably one of my least cited papers and one of the most papers I'm proudest of, but basically about how to be able to create these fields. And then I wanted to build this microscope, and of course I tried to get Harold to build it with me, but he said he didn't really want to do it because it was chewing my cud, and he wanted to do something on his own, right? But he was going to help me to find a place where I could do it. And so one of the places we went to was Florida State University, where the director of the High Magnetic Field Lab was actually a friend of ours from Bell. And there was a weird guy there who was doing live cell imaging on the side called Mike Davidson. And he told us about a new type of fluorescent protein that had just recently been published. In a normal fluorescent protein, if you shine green light on it, or shine blue light on it, then it glows green. But in these new photoactivated ones, you shine blue light on it, nothing happens. Unless you first shine purple light on it, and then it activates these molecules so that then when you shine blue light on it, it glows green. So they're photoactivated fluorescent proteins. And Harold and I were sitting in the airport in Tallahassee getting ready to leave, and we said, my God, this is exactly the missing link to make that idea that I published in my first round unemployment work. Because what we could do is we could turn that violet light down so low that only a few molecules get photoactivated. Then statistically, they're likely going to be separated by more than the diffraction limit, so their fuzzy blobs don't overlap. Then you can localize those, turn those molecules off, turn on another subset, do it again and again and again. So it was the idea I had 10 years earlier, but now with time as this isolating characteristic. And we said, oh my God, this is so ridiculously simple. Why hasn't anybody done this? So we were terrified. It's like, so we quick look at literature. No, nobody's done this. Well, God damn it, we got to do it. I mean, it's just a compulsion. It's so easy. And we raced because we were concerned. We were right to be concerned. There were other groups right on our heels. So the good thing is that we had a couple of advantages. And the first one is that when I left Bell, I told them all to go to hell because I was frustrated. But when Harold left Bell, he was able to take all of his equipment with him because he's a lot smarter than I. And so we took all that out of the storage shed and put about 25K each of our own money in it. And the other advantage was that we could build it in the living room instead of in the garage like Jobs and Wozniak, because Harold wasn't married, so there was nobody to complain if we did it there. And we built it in a couple months, and then we shipped it to NIH because I had made connections with Jennifer Lippincott-Schwartz at NIH, who, with George Patterson, were the inventors of this photoactivated fluorescent protein. And so we took it to their lab, and in another month or two, we had images that looked like this. So this is a slice through a multivesicular body, sort of a garbage dump in the cell. This is the regular diffraction limited image. This is the palm image. Or to better appreciate, you go from this type of resolution to this with a silly little microscope you can build on a living room floor. So this immediately took off because it's easy. One of the things academics love is things that are easy. So everybody loved things that are easy. This was easy. So we took that and ran with it. So at the same time, 2005 was just a crazy year. Not only did we come up with Palm, I also got hooked up with Jerry Rubin, who was this guy who worked for this outfit I had never heard of called Howard Hughes Medical Institute, who wanted to recapitulate Bell Labs in a biological sense at something called Genelia Farm. And so I got hooked up, and I was one of the first hires. And then I said, you got to hire this Harold guy too, and so they did. And so that was a great place for a number of years to work. So we did a number of applications, and the field continued to grow along, but very quickly it was the same story I had with the near field days, where the signal stays about the same and the noise goes to infinity. And there's a lot of problems with super resolution. The first one is the vast majority of what we've learned has been on chemically fixed cells, but the EM community is known for 80 years now that fixatives destroy ultrastructure. So what is it that you're seeing something at high resolution, but it's not exactly what used to exist. So the way the electron microscopists dealt with this was by doing something called high pressure freeze, where you slam an anvil down on a sample in aqueous solution that's cold. In the high pressure and the low temperature causes the ice not to crystallize but form a vitreous form, which then encapsulates and preserves the ultrastructure. You can then substitute in a resin and heavy metals to do staining. As you can see in this case, this is from Harold's FibSem microscope. You can see that with the cryofixation, You maintain all of the structure, for example, of the chromatin in the nucleus, which is completely disruptive if you use chemical fixation. But now you have a sample that's cold, and you want to look at that. Now that means you need to have a cryogenic palm microscope. And so the high pressure frozen, you end up getting something like this, but it takes a microscope that looks like this, which doesn't look much like a microscope. It looks like a physics experiment, which basically it is. So it would run down to 4 degrees Kelvin. We put in these frozen samples. We do the super resolution localization imaging, and then we take that sample out, we resin embed it, put it in Harold's crazy microscope. Harold is now a god at Janelia because of this microscope he developed, which is basically taking a block of tissue, using a focused ion beam to mill away a few nanometers at a time, image the top surface, and do it again to get 3D EM images at 4 nanometer voxel sizes throughout now whole fly brains and working towards whole mouse brains. So we could use that along and do a correlative now with the super resolution, which allows you to get the specific protein-specific contrast you get from super resolution with the global contrast you get from the heavy metal staining from the EM, as you can see here. So that immediately revealed a number of surprises. First was in this top case, when neurons come together, we discovered a certain complex of molecules that create a web-like network between these two guys at their plasma membranes. And furthermore, this network has a certain protein which create corrugations in the membrane, which you'll see when this slices through. We also saw that in the EM community, it's long been believed that when you look at the nucleus, that these dark regions are parts of the chromatin which is inaccessible for transcription, and the clear regions are accessible for transcription, euchromatin versus heterochromatin. But when we put on fluorescent markers of transcriptional accessibility, we found they didn't correlate with those EM things. So it basically puts a question on the whole idea of exactly how they interpreted those EM images in terms of accessibility for transcription. So all of that is fine, but the other problem with all of this that we've done is because the technique has been so slow to be able to look at all of those molecules a few at a time, that the easiest way to do it is with chemical fixation or high pressure freeze. But I heard a talk by another microscopist around 2008 when I was starting to get as sick of palm as I had gotten sick of near field back in '93. And what this guy, Scott Fraser, said is, you know, biology is about trying to understand the rules of the game by which molecules self-assemble to create life. And if you had even the highest resolution TV of a game and all you had was still pictures from that, it would be impossible to understand the rules of the football game from seeing the quarterback going back to throw a pass, the cheerleaders making a pyramid on the sideline, and the halfback fumbling a ball. But if you could just watch the game or go to the game and watch it, then the rules become far more obvious. And when I heard that talk, I said, shit, I've been looking at dead stuff my entire life. I am done with that. I'm now only going to look at living things. And so I wanted to figure out what to do live. And the other problem, too, is, you know, animation is what defines life. It's the thing that separates it from this water bottle or this computer, okay? So if you don't study that animation, you're never going to understand the system. And stochasticity is fundamental to everything about life. And so you have to come to grips with natural variability. You have to see that variability live, and you have to see it with high N, high number of samples to be able to get a sense of the breadth of that variability. So how can you possibly understand life without looking at it live? And yet still today, the vast majority of biologists look at things from those 3 old worldviews of biochemistry, molecular biology, and structural biology, which are about taking life apart and looking at its parts and not looking at its totality. And I say, this is where optics can really excel. So and there's a couple ways in which this has succeeded. So with palm-type techniques, we don't need to look at a fixed cell if we just want to see how cells move around, or how molecules move around in the cell. So we can photoactivate subsets of molecules and look at their diffusion. And for example, with molecules that are non-sticky in the plasma membrane, they move all over the place. With molecules that like to bind together to create virus-like particles, they cluster together. And you can see that immediately by looking at those types of things. So a good example of applying these sort of single molecule techniques is this experiment done by a collaborator at Janelia. So what you're seeing here are two different nuclei from two different neurons. The left one has the wild type Huntington protein that's involved in the Huntington's disease. The right one has the mutation that causes the disease. And so you can see immediately that while the protein aggregates in both cases, they form large and stable aggregates with the mutation. Now that's been known for many years, but and there's a lot of theories about why these aggregates then result in the phenotypes of the disease. But one novel possibility that was shown by this experiment is it was a two-color experiment where these little purple fireflies are transcription factor molecules, and they get bound up like glue inside of those aggregates. That means that they can't make it then to the DNA to be able to then eventually be able to create RNA. So that means that the proteins that are transcribed that are needed by those transcription factors to transcribe certain RNAs and result in certain proteins are not happening. So there's a down regulation of certain proteins. So that possibly could be one mechanism by which these aggregates do it. So again, it's by looking that you start to get these hypotheses. And this is a really powerful tool to do that. In fact, let me turn that off. Another example of this is, again, transcription. So you start with DNA. In order to copy DNA, there's an intermediate messenger RNA which gets transcribed on the DNA. Then it goes over into the ER and the ribosomes to create proteins. So this is an example of this polymerase, which is scooting along the DNA and spitting out this ribbon of yellow RNA in this case. So over time, biochemists have figured out which proteins interact with which other proteins to create a point, a spot called a transcription initiation complex, to recruit that RNA, to recruit the polymerase, I'm sorry. And so they had in their mind that there would be this stable transcription initiation complex which comes together over the course of many minutes or an hour. But when we started to look at the individual molecules involved in that complex, we saw that they were only binding for seconds. And so how the hell does this happen then? Well, instead, the model that emerged is there's not just one copy near the start codon for a particular gene, but multiple copies of each transcription factor, and they hover like bees near a beehive in order to come together. But what causes them to hover together? Why would they want to do that? Why wouldn't they just go all over the place? Well, they have parts of them that are intrinsically disordered regions, and these act as sticky points to have certain different types of proteins that have higher binding to certain other types of proteins to come together in order to do this. And in fact, when you snip those off, you actually change, for example, a childhood cancer like Ewing sarcoma, which is dependent upon a mutation that causes these IDRs to go on. You cut them off, and then the phenotype and single cells go away. Now, we can't cut them off in a human, but it points to the possibility of, could we do something to inactivate by chemicals what these IDRs are doing in order to recover a non-disease phenotype? And so that led 4 of us to start a drug discovery company which uses single molecule tracking and then putting in different compounds to see how the kinetics of single molecule binding and diffusion is affected by different drug compounds. And so that company we started in 2019. This company has been amazing in what they've been able to learn. So they're still a long way from a clinical outcome out of it, but they know more about how molecules move and how molecules interact with other molecules than anyone else on Earth by a mile. There's roomfuls of single molecule microscopes running 24/7, creating a petabyte of data every day, which is reduced to understand how 300,000 different compounds every month are tested to see how they affect the motions of different proteins that are involved in different types of diseases. So if they're, I still to this day don't know if if super resolution is worthy of a Nobel. But if that company actually comes up with a good approach to drug discovery, I might change my mind. So jury's still out on that. So the moral of the story so far is that, the focus on resolution, which usually means dead stuff, is really just looking at one metric that's important in a microscope. But for biology, there are many important metrics. Are you hurting the cell? Is it fast enough to be able to see the dynamics you want? Is there limitations to how deep you can image inside of a specimen? And so there's no such thing as a free lunch. There's always trade-offs. And the guy who understood this earlier than any of us was Mats Gustafsson, who, as a grad student and postdoc at UCSF, developed a technology called structured illumination, where instead, a different super resolution method, where instead of imaging the sample uniformly with laser light, instead you put on a grading pattern of laser light, which can demodulate information in the sample that's beyond the diffraction limit to a frequency band that's within the diffraction limit that you can see. Because the pattern can only go up to the diffraction limit itself, the applied pattern, it only gives you a 2x gain in its normal state. But even 2x, now it means I only need 9 images as I spin this around in order to distill a single super resolution image versus thousands of images with Palm. And so it's not as high resolution, but those are the trade-offs, but it's twice the diffraction limit, and you can do live cell imaging at high speed. Here's 10 frames a second, and it's less invasive because you're not asking for as much fluorescence from individual molecules per unit time. So this is looking at a T cell, part of the adaptive immune system, plopped against a cover slip with antigens on it to provoke the immunological response. And so you get this interface between the T cell and its target with this characteristic actin network that they can see in this inflowing from the outside. You can go to slightly higher resolution if you really tweak the NA of the microscope in what's known as total internal reflection, so you can see how myosin, a protein, creates these dumbbells which then stitch together actin filaments to pull them together to squeeze the cell from behind and propel it along. And if you do, the other nice thing about SIM is it's easy to do multicolor imaging, which is harder with single molecule. And so this is an example of looking at 3 different organelles at once, and it reveals that the organelles are not like this textbook picture of here's some microtubules, here's some mito floating over here, here's a lysosome over here, everything interacts with everything else. There's things happening all the time. The ER is remodeled by the microtubules. The lysosomes are actively transported by the microtubules, bump into the ER, remodel the ER. There's contacts and probably biochemical events happening at all of these points, and that you can see that through the SIM. So I'm going to skip this slide in the interest of time, but there's lots of actual biology that we've been able to contribute by SIM. I would say that if there's a hierarchy of, again, to this day, many people say, why aren't you just doing single molecule super resolution? It's got the highest. Resolution's not the only metric. I've worked with a lot of tools, and in my hierarchy, basically I've found that in terms of biological discovery, that generally the lower the resolution tool, the more biological discovery I've had with it. Okay, so SIM is in the middle, and we've had a lot of interesting applications with that. But SIM, as it was done most of the time, you can do SIM in 3D, and there's benefits to it. But the problem is because every raw image that's distilled into a super resolution image by SIM is itself a wide field microscope image. If the cell's more than a few microns thick, there's a lot of out of focus stuff which defeats your ability to do the computational reconstruction that you have to do. And yet, the cell is a 3-dimensional dynamic system. Every living thing is a pocket of reduced entropy. Matter and energy is flowing through it continuously, and the only way we're going to understand such systems is by looking at them with high resolution across all 4 dimensions of space and time at the same time. So how can we do that? So when I got frustrated, first with Palm and somewhat with SIM, I wanted to find out a way to contribute, and again, also inspired by Scott Frazier's talk about the football game. So the good news is there was an opportunity because the conventional microscopes most biologists use to look in 3D have a lot of room for improvement. I already mentioned the wide field microscope with its out of focus problem. The confocal microscope uses a pinhole to collect light only from the focal region and throw out the light from the regions above and below that are illuminated that are out of focus. That's fine, but you've just illuminated those areas. That means you're creating damage in those areas. That means you're bleaching the fluorophores in those areas, and you only have a limited photon budget. So in my opinion, the other really transformative idea introduced to microscopy in this century was when Ernst Stelzer reintroduced this 100-year-old idea called plane illumination, where instead of bringing your light from above, you use a second, in this case, for example, a cylindrical lens from the side, to create a sheet of light which is coincident with the focal plane of the detection objective. And now you're only illuminating one plane. So the areas above and below, they're not excited at all. There's no out of focus light, there's no bleaching, there's no toxicity. Because you're illuminating a whole plane at once instead of a point, you don't need to have such high intensity, but you can have lower intensity because they're all emitting photons in parallel. So it can be a lot less toxic. That's been transformative for understanding embryogenesis at single cell resolution. But it's generally limited by the fact that the normal technique uses a standard Gaussian beam, which has a certain relationship between how thick the light sheet is and how far it can extend. And so our thought was, well, in the 80s, at the same time the near field was happening, there was some buzz about non-diffracting beams called Bessel beams, where instead of illuminating the back of your objective with a round illumination, you use a ring. You now have two knobs, the thickness of the ring and its diameter, to decouple how long it is from how thick that beam is. If you could sweep that beam back and forth in a plane, you mimic a light sheet that's much thinner, so that's then Bessel beam plane illumination. We then eventually realized, after several papers with that, as we tried to work on arrays of Bessel beams to make that process faster and less invasive, that there were certain periods at which we were able to strip out a lot of the out-of-focus light from the Bessel beams. And I realized that these magic periods were periods predicted by that optical lattice theory that I had done and completely abandoned when we did Palm, but turned out to be perfect for being able to predict what type of light sheets to create for this now lattice light sheet microscope. So this is the lattice light sheet microscope. I've worked with probably about 100 different groups at Janelia over about 6 or 7 years with this microscope. And I'm proud to have won the Nobel Prize for Palm, although I'm not still quite sure it's good for. But I know in my bones that this microscope will be the most impactful microscope I've ever made. Because with those 100 people, I have never felt more like Galileo with his telescope than I have with this microscope. Because everybody who comes into the lab and tries this out, these are people who have been working on these systems for 20 years, and they see them in a way they have never seen them, and they're just agog. And they say, it's amazing what they can see. You know, like this little crazy lightning bolt in a stick to steel you're about to see. You know, the guy first saw that and he said, what's that? And I said, I don't know, you're the biologist, you tell us, you know. But so everybody goes away with 10 terabytes of data and big smiles on their faces. And then a month later, they come crying to us saying they have no idea what to do with 10 terabytes of data. And you laugh, but sadly, that is the Achilles heel. The ability to, it's not the microscope anymore. It's the ability to analyze and make sense of the data, which has been completely defeating so far, and I'm going to get more into that topic if I don't run out of time. So I'm going to skip over the applications we've done with lattice, but they've been voluminous. Too many papers to count. Again, it's been an amazing tool of discovery for us. Lattice light sheet, of course, now we get into more modern age of my stuff. So it's been obvious to me for a long time, from both the Palm days and then certainly the SIM days, that cells did not evolve on cover slips. It's an incredibly artificial environment. I've soaked up enough biology from my collaborators to know that the phenotypes that exist inside of cells. A liver cell is different than an eye cell. Why? Because they live in different environments. And so they all have the same DNA, but they're different because the phenotypes we observe are the result of gene expression. And gene expression is controlled by environment. And that environment comes from exterior signals. So if I put something in an artificial environment, I'm going to get artificial phenotypes. So it doesn't matter how good my microscope is, I'm still not seeing reality. So I have to meet biology on its own terms. I need to look at cells in the living organism in which it evolved. The problem is, as you can see, there's a loss of resolution as we start to go into multicellular specimens. And the reason for that is because cells are not homogeneous. They have cytosol, they have nuclei, they have mitochondria. All these things have different refractive indices. And trying to look inside of a living biological specimen, even if it's transparent, is like trying to look into a bag of glass marbles, okay? And the light eventually gets scrambled, or it's like trying to look at the cars in front of you when you haven't turned on your windshield wipers. So how do you deal with that? Well, in my talk this afternoon, the main take-home message is going to be that microscopists are the retarded stepchildren of astronomers because we steal everything from them 50 years after they invent it first. And so this is going to be another example. So the atmosphere does the same thing. If you want to see anything out in outer space, you can build the biggest telescope you want, but if the atmosphere is scrambling the light, you're sunk. So there's two ways around that problem. The first is to put your telescope above the atmosphere and get rid of the aberration. That's what the Hubble Space Telescope does, right? And that's been an amazing tool. This is an example of the Crab Nebula with Hubble. This is it with the Hale telescope, which is still the biggest telescope on Earth. But it doesn't have adaptive, or didn't have adaptive optics at this time. So this mirror is half the size of this mirror, okay? But the resolution is much better because it's adaptively optically corrected, okay? So that's great. Can we do, or not adapt, that one's in space, so there's no aberration. So is there an analogy in microscopy? The analogy is called chemical clearing. You can put different chemical cocktails inside a chemically fixed tissue in order to homogenize the refractive index. And there's a lot of these, but one of the more clever ones was developed by Ed Boyden at MIT called expansion microscopy. So in this case, what you do is you fix the specimen, you put in this polymer inside of the thing to homogenize it. You then do some chemistry to link the fluorophores that were in your biological specimen to the polymer. You then bring in a protease to digest away all the biological stuff, so all that's left is this phantom of where the fluorophores were. Then you change the osmotic balance that this polymer is in, and the polymer then swells 4x. This is not a magnified image of this brain. This is at the same magnification, but the brain actually physically swells like some science fiction movie or something, right? And so in this case expands 4x. So now your diffraction limited microscope is a super resolution microscope. Unfortunately, it's fixed, it's dead, all that stuff I said, but at the same time, pretty cool. In fact, I didn't believe it. I looked at some of the data in their paper and I said, that looks like shit, okay? And then they knew about our lattice light sheet. The problem they were having is when they're trying to go into specimens this big, because now they're big, they're expanded, is that the bleaching was killing them as they were going deeper. And they didn't have the resolution they wanted either. And so they knew about the lattice light sheet and they wanted to bring it. And I said, sure, come on by and we'll try it. And I'm just going to rub their nose into all the artifacts I saw. And then we start looking at something like this one on the bottom of a section from the mouse brain. And I see everything exactly as it's supposed to look at higher resolution. You could have knocked me down with a feather. And I said, okay, well, I owe it to you guys. So we did a paper together on this, and it's been pretty cool, I have to say. It's dead, but it's pretty cool. But still, I want to do live. And so for that, astronomers come to the rescue again. The problem with the Hubble telescope is there's a limit to how big a mirror you can put in your payload bay. And even then, if like in the Hubble, you grind your mirror wrong, you're really screwed unless you do $1 billion service call, which is what they had to do. But with your microscope on the ground, you can build a bigger mirror. but you have to get rid of the aberrations. So how do you do that? Well, what you do is you shine a laser up into the stratosphere to excite sodium atoms to make a very bright artificial star. And that artificial star is near the extremely dim astronomical object you want to see. The light from both of them come back down into your telescope. They're distorted in exactly the same way, the light. So you bounce them off a mirror that can change the shape. Then you peel off the light that comes from the bright artificial star into a sensor that can look at that distorted shape and deduce from that distorted shape exactly what the aberration was. You then put that in a computer in a closed loop to change the shape of the mirror to exactly cancel that distortion. And then your dim astronomical object is back at diffraction limited resolution. It's like your telescope was in vacuum all of a sudden. Cool. Okay. So you go from Saturn looking like this to looking like this. They were actually able to show, looking at the center of the galaxy, that there's a supermassive black hole around which various stars orbit. That led to Nobel Prize in Physics a couple years ago. And even better than the Hubble Space Telescope now, because it's a bigger mirror, because it's on Earth and doesn't have to fit in a payload bay. So it's awesome, really awesome. So we can do this exact same trip with microscopy. What we do is we use what's known as a two-photon excitation. So that way there isn't a cone of light, but it's only where it's super intense that you get a single spot of light of fluorescence. Use that as our guide star that goes back into our control loop. And you go from looking in the hindbrain of a zebrafish, from looking like this, low signal, poor resolution, turn on the AO, bing, there you get it, just as good as if it were diffraction limited. So it's a great tool. This is a couple examples. The one on the right, you're looking at the soma of a single neuron in the hindbrain with some processes around it. These are mitochondria, actually. Turn on the AO, you see the mitochondria. You do a deconvolution, and it's just like you were looking at a cell on a cover slip instead of deep in the organism. Here you can see that we can, the problem with AO is that the aberration changes as you go through different material, so you have to correct it again and again and again as you move along. So that's called the isoplanatic patch. This is turning off the adaptive optics, and again here turning it back on, but you can cover large fields by doing multiple corrections across the large fields. This is an example. Yeah, it's doing this again. Eventually this computer is about to crash and I'm going to have to start it up, watch. But anyway, so yeah, this is an example of looking at now, we eventually married then, so we have two pieces of the puzzle. We have lattice light sheet, which is non-invasive, fast, reasonably high, but not super resolution. And we have ability to now take that into a multicellular context to look at cells in the physiological environment in which they evolve, which was my goal. So we put those pieces together. This is an example of that. This is looking at the development of the spinal cord in a live zebrafish. So those little railroad tracky things you're seeing are axons, which are actually growing and maturing as they move along. We found out the axons that go from head to tail have a different characteristic shape than the axons that go from dorsal to ventral. This is now showing what happens to cells, not in a cover slip, but again, in a real organism. This is in the ear of a zebrafish. This is a neutrophil, so part of your innate immune system, that's scooping up this bait we put in, which is some sugar particles. And so you can see how crazy they are in their morphologies and their ability to move. Completely different form and completely different speeds, completely different dynamics than a neutrophil on a cover slip. And so you can see how complicated living systems are. You're made of 37 trillion cells of thousands of different cell types. Here you see just a few dozen of the cell types that happen in a fish, and each one of your cells has 10 billion molecules in it. So this is the biggest mystery in the universe for us, is to understand that level of complexity and how it maintains how it developed and how it maintains itself. It's beautiful and mysterious to this day. I'll skip this stuff, but we did some stuff looking at how cancer cells get out of blood vessels and into the body. So that's been my favorite microscope so far by far, the AO lattice light sheet. The problem with it is that the initial one, because you have to have adaptive optic correction on the light sheet because it goes in one pathway, and another adaptive optic correction on the detection because it goes in a perpendicular pathway. So it's like 3 microscopes in one, filled a 10-foot optical table and cost $1 million. And a big part of my group has been about trying to disseminate our technology to other groups. Nobody was going to want to rebuild that thing. So we had to build a second generation version. And we also realized in doing that, we realized that once we have all the components we need to do adaptive optics and to do lattice light sheet, it has all the components that are in every microscope that's basically been invented since that revolution started in 1980. So why not make it a Swiss Army knife that can do confocal, wide field, structural illumination, single molecule, in one box? And so that's what this is. It's called Mosaic. So it's this multi-functional adaptive optic microscope in any one of those modes. So it can do phase imaging to look at. The advantage of phase imaging is it doesn't provide protein-specific contrast, but it's non-invasive and gives you overall contrast to cell morphologies. We can do single molecule localization in 3D. The instrument is stable enough to do that over fairly wide fields of view. We can do 2-photon imaging in the mouse brain, doing adaptive optics, scrolling back and forth between AO on and AO off, or do imaging of functional activity of neurons in the brain with and without AO and getting better signal with the AO. We can also combine structured illumination with lattice light sheet to do a higher resolution version of lattice light sheet. In this case, again, looking at a field of cells. But the big thing about the Mosaic is it has a very large imaging chamber and a very high imaging speed. So this is looking at nearly a millimeter square of several thousand molecules, or not molecules, cells in 4 dimensions dividing over 24 hours. So we're going through many, many, many division cycles in this case. The original data is about 100 terabytes of data. The microscope takes data again at 4 terabytes per hour. So every week, one microscope produces half a petabyte of data. This is where we're stuck, so I'll get into that in a little bit. But the advantage of this microscope is it allows you, and we have the intelligence now to do this, is to look for rare events. It isn't in this movie, but if you look in this, you'll see one example where a cell doesn't divide into 2, it divides into 3. And so there's all sorts of, you can also then follow the trajectories of over the time of those daughter cells and what happens to them, or speed the movie backwards and find out what were the precursors to that. So of course that then leaves us with the problem of how to handle data at that scale. Again, I am not a data guy, but sadly I've had to lead a bunch of people who are data guys because this has become the bottleneck. And the bottleneck to the point where it was untenable to rely on even Berkeley's computing center, so we had to develop our own cluster inside our own lab. The Mosaic cost $800,000, but to be able to analyze the data off the Mosaic required a compute system that cost 3 times as much. But with that, plus a lot of talented guys who are computer scientists who found out ways of doing distributed computing of all of the data we get across many, many GPU nodes inside of that cluster. We're able to do all the pre-processing work on that stuff in real time, compress it to a reasonable size, and store it, but we still don't have good ways to visualize and deal with it. So in the past, in the 2 papers prior to that where we did massive amounts of data, which was the expansion microscopy paper I told you about, and then the lattice with the adaptive optics I told you about, my buddy Gokul, who's my right-hand man at Berkeley, did the analysis by just normal analytical tools, extracted many insights about both the mouse brain and in terms of morphogenesis in the zebrafish eye. But it took, each paper took 3 million CPU hours on the Genelia cluster, and they have a pretty capable cluster. And I can't clone Gokuls. I wish I could, but I can't. And so this is not a scalable answer, and this is still only a tiny fraction of the data we have. So we need a different solution. And the problem gets worse because, as I said, the diversity of things that happen in living systems is immense. There are so many types of things happening at the same time, from immune cells to the anchoring of epithelial cells and how they interact with the lower layers, how the mesenchyme develops itself. And all of this is happening simultaneously. All of it is different. And if you write a custom code to analyze one type of process, well, there's 10 to the 9th other processes going on you'd have to write a custom code for as well. So you're kind of sunk. So what do you do? Okay. So this is where we're at, and this is where I'm stuck, and this is where I've started in the last few months to make a pitch for where we have to go. So the problem is that the type of 5D with color of the interactions inside of living tissue, it's just too incomprehensibly complex for you to get any understanding of what is going on inside in that small block of tissue, okay? So the hope is that, well, you think about what's happened with ChatGPT, right, is that we have now, the big step forward with ChatGPT 4 was by using these transformer networks that use self-attention to the sequence of words in a sentence to be able to then make leaps forward and being able to construct sentences. So that same spatial temporal priors that are used in transformer architecture, well, spatial temporal is what we have out the wazoo here. So transformer architectures are probably the right way to bring machine learning in to help with the analysis of this type of data, okay? In fact, Google a couple years ago actually applied this to vision in 2D, where they were able to then they took basically 300 million images scraped off the web, trained a transformer model to be able to identify whether there's a dog in that image or a cat in the image or whatever, and get that to work. So that was the state of the art like 2 years ago. Based off of that transformer model, Meta then took that and then applied it to the job of segmentation. Now that's the very first thing you have to do in a multicellular specimen if you want to understand subcellular dynamics, is where does one cell begin and when does it end? So they have an ability to do this in 2D. It took, again, lots of images, fair amount of time. Over a year on a single H100 GPU, you probably know that NVIDIA had their big conference last week where now the B100 is coming out, which is considerably more performative than the H100. And so we're relying on this virtuous cycle of what's happening in GPU hardware with the virtuous cycle of what's happening in transformer and other networks in order to be able to do what we want. So the first step, like I said, is segmentation. This is an example of us segmenting that tissue just like we'd like to do, but it was done by GoCool with analytical methods with a lot of hand annotation in the end for where it screwed up. So what we need is a way for machines to be able to do this quickly and robustly, and AI is probably the path forward to do that. The good thing is that for this path of segmentation, there's an example in biology where this is done automatically by something called Brainbow. So you use green fluorescent protein and its variants to express different colors in different cells randomly, and so then they light up. We could use this to train an AI model to do this segmentation task. But then once you've got it segmented, you're just getting started, that's just the ante. Then you want to do all sorts of analysis of the subcellular organelles, where they contact and so forth. And even once you get classification of cell type, cell state, their interactions, how they remodel, you still have to access that, even if that's all figured out by the AI. And so the final thing is because even physicists won't be able to get into that data, and biologists sure won't, you need a large language model to have a conversational interface where the biologist could say, okay, when a neutrophil is trying to extravasate through a blood vessel to go to a tumor, what happens, how does the different organelles have to remodel themselves to make it through that small gap? And you could ask the question that way. And in the future, the AI would come back and say, here's 5 examples we have in the database of exactly that happening. Here's exactly what happened to this organelle in terms of how it had to fragment itself and then reassemble on the other side through the interface. That's the way we would really push the needle forward. Okay, that would be the dream, is to try to get to that. And that's what I'm trying to advocate now. The bad news is that I've always been a guy who's wanted to work alone or with my group. Everything that you see here, my group has rarely been bigger than 3 people. I do not believe in big labs. I don't know if you guys know something called Price's Law, that half of the output is done by square root of the number of people in the organization. It's really true. It extends over many, many things. So I feel like I've been productive by having a very small team. But I'm afraid that this project is something that's kind of more like building in the Hale telescope, that it requires a lot of expertise in a lot of centralized place. Whether I find other people to share that vision and pony up the money to do it, I don't know, but that would be the goal. So let me conclude by saying biochemistry, molecular biology, structural biology, we've learned a lot from those tools, okay? But my belief is that if we can overcome this data problem and really be able to see what's happening at all length scales inside of living tissue, that this will unleash a new revolution in our understanding of biology, far more profound than even what we learned in the 20th century. With that, thank you very much.